Good afternoon. My name is Thomas Harris Jr., and I will be presenting today on the bacterial species Azotobacter vinelandii. I primarily chose this topic because of an interest that was sparked in me to learn more about photosynthesis and the various cycles and mechanisms that are involved in that process in particular. I came across some interesting information on nitrogen fixing bacteria, which I will go into greater detail about throughout this presentation. So the points that we are going to discuss involving Azotobacter vinelandii include its taxonomic classification, its physical and behavioral features, the habitat that it lives in and its distribution, the evolutionary adaptations, and the two concepts we have discussed, commensalism and survivorship. A. vinelandii are rod-shaped aerobic bacteria that was first isolated in the soil in Vineland, New Jersey. They reproduce asexually, meaning that they reproduce using basic cell divisions, during which they replicate all parts of the cell, and then the cell splits itself into two parts. A new cell is created and is therefore able to repeat that process. These bacteria thrive in non-acidic conditions, which mainly include desert and Arctic soils. However, they have the ability to survive in a variety of intense living conditions due to major evolutionary adaptations that have supported the survival of this organism. And I will discuss more about that a little later. These organisms are free living and are able to carry out carbon fixation while growing aerobically. Free living bacteria means that they don't need to create symbiotic relationships with plants to survive and replicate. Azotobacter contains 6 different species and belongs to the Pseudomodidaceae family. Researchers determined that Azotobacter species is very closely related to the Pseudomonas and have made note that although Pseudomonas do not have the nitrogen fixation as being one of their main characteristics, these two genera are closely related in structure, phenotype, and morphology. The diversity of phenotypic characteristics of the genera, including morphology and nitrogen fixing capability, could be due to the adaptive bacterial transmission of genes. However, the extent of which bacteria show phenotypic expressions due to this transfer is not very clear. Research is currently being done to further analyze the synonymy of Azotobacter and Pseudomonas. Genus Azotobacter are aerobic organisms, which mean they require oxygen to grow and reproduce asexually. They live in the soil and are rod-shaped and negative in gram staining and procedure, incapable of directed movement via a flagellum, and they are very large in size for bacteria. They also are able to produce colored pigments in soil. In regards to the habitat that Azotobacter vinelandii bacteria thrive in, you'll most likely find this species in a non-acidic soil located in the desert and the Arctic areas of the world. If you take a look at the image, this shows the solar radiation spectrum, and Azotobacter vinelandii are able to live in intense conditions due to the mechanisms and adaptations that we will discuss later in this presentation. The map on the screen indicates the major biomes of the world, and if you would focus your attention to the areas circled in red, they show the extreme living conditions of the world that you could possibly find Azotobacter vinelandii living in. Azotobacter are able to form cysts, or resting cells that are surrounded by protective coating. Cysts are important to the survival of these organisms due to the fact that they are promoting resistance to drying out or desiccation. Microorganisms are unable to grow and divide when desiccated, so this feature is very beneficial to Azotobacter and allow them to, or the ability to survive in harsh conditions. So that includes the tundra, where there is intense cold, and dry desert areas where there is intense heat. When in a cyst form, these organisms are unable to fix nitrogen. Another evolutionary adaptation associated with A. vinelandii includes slime secretion. Slime naturally retains moisture, and when the bacteria secretes, this material, it is used to surround each bacterium and take any water that is nearby. This is very useful in particularly dry climates where A. Vinelandii may be found. In terms of Azotobacter survivorship, like I mentioned before, these organisms are generally found in slightly alkaline soils, and are well known to produce large quantities of extracellular polysaccharides (EPS). These substances are used as a protection for the Azotobacter as they stop toxic metal ions from penetrating inside the cell. The second concept being discussed is commensalism, which can be defined as an association between two organisms in which one benefits and the other derives neither benefit nor harm. So, Azotobacter vinelandii has the ability to fix nitrogen under aerobic conditions. In other words, these organisms utilize the enzyme nitrogenase to catalyze the conversion of atmospheric nitrogen into to ammonia NH3. Nitrogen is a critical limiting element for plant growth and production. Therefore, agriculture meets its required amounts of nitrogen by forming symbiotic relationships with soil bacteria, such as Azotobacter vinelandii. This fact alone makes this genus of bacteria especially favored for agriculture purpose. So why is A. vinelandii so important for our survival? The nitrogen cycle is one of the most essential biochemical processes on Earth, and understanding how this cycle works can help human beings make better decisions about what crops to grow and where to grow them, which will help ensure there is an adequate supply of food. The work of A. vinelandii plays a crucial role in maintaining productive and healthy ecosystems, with neither too much nor too little nitrogen, being that all living material on Earth is limited by the availability of nitrogen that is available.